Welcome back to Dr. Liu's VCE Biology. Today's topic is the relationship between nucleic acids and proteins, as presented in the 2025 VCE Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. Based on the diagram provided, the correct answer is D. Protein X does not undergo further modifications after leaving the rough endoplasmic reticulum. Protein Y, the conventional pathway, you can see protein Y, black dot, moving from the rough endoplasmic reticulum, RER, into a vesicle, then entering the Golgi apparatus, the stack of flattened sacs. In the Golgi, proteins are typically modified, sorted, and packaged before being sent to the plasma membrane. Protein X, the unconventional pathway protein X, gray dot, leaves the RER and moves directly to the plasma membrane in a vesicle, bypassing the Golgi apparatus entirely. A. Incorrect. The diagram explicitly shows protein X reaching the membrane without entering the Golgi. B. Incorrect. Protein Y is shown passing through the Golgi apparatus, where modifications definitely occur. C. Incorrect. Both proteins are shown inside small circles, which represent vesicles used for transport. D. Correct. Since the Golgi apparatus is the primary site for post-translational modification, like glycosylation, by bypassing it, protein X avoids those further finishing touches. Because the mRNA is complementary to the DNA, and the tRNA is complementary to the mRNA, the tRNA anticodon will look exactly like the original DNA triplet, with one major exception: all Ts must be US DNA triplet, GC at RNA. Anticodon, GCU. Correct answer, D. RNA polymerase reaches a termination sequence on the DNA template. This question tests where the boundary of transcription lies. Transcription is strictly the process of copying DNA into RNA. YD is correct. Transcription is carried out by the enzyme RNA polymerase. When this enzyme hits a specific stop sign termination sequence on the DNA, it detaches, and the primary RNA transcript is complete. Why the others are wrong? A. Removing introns is part of RNA processing, or splicing, which happens after the initial transcription is done. B and C. These events involve ribosomes, which means they are part of translation, making the protein, not transcription. Four different sections of DNA are described below. Question contains exons and introns, and requires splicing to form mature mRNA. R. A cluster of genes under the control of a single promoter, transcribed together into mRNA. S. Has a promoter region for RNA polymerase binding and does not have introns. T. Codes for proteins that control the expression of other genes. First, look at question. The giveaway is exons and introns. Since only eukaryotic genes have those non-coding intron interruptions that need to be spliced out, Q must be the eukaryotic structural gene. Next, look at R. It mentions a cluster of genes under a single promoter. That is the textbook definition of an operon, like the lac operon in bacteria, where several genes are turned on or off at the same time. Then, look at S. This one has a promoter but does not have introns. This is the classic setup for a prokaryotic gene. It's streamlined and ready to go without any fancy splicing. Finally, look at T. It says it codes for proteins that control the expression of other genes. That makes it a regulatory gene. It's essentially the boss gene that tells other genes when to wake up or stay quiet. So, option C is correct. 1. From gene to protein transcription ends when RNA polymerase reaches a termination sequence on DNA-RNA processing, splicing, removes introns. Mature mRNA translation occurs at ribosomes using tRNA anticodons 2. DNA left and right arrow, RNA base pairing rule mRNA is complementary to DNA. tRNA anticodon is complementary to mRNA. Result: tRNA anticodon matches DNA triplet except TU. Example: DNA GCA tRNA GCU3. Protein transport pathways. Conventional pathway. RER Golgi plasma membrane post-translational modification occurs. Unconventional pathway. RER plasma membrane Golgi bypassed equals no further modification. 4. Types of genes. Prokaryotic gene, promoter present, no introns. Operon, cluster of genes under one promoter. Eukaryotic structural gene, exons plus introns, requires splicing. Regulatory gene, controls expression of other genes. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.